development side of things, and library prep, preparing a DNA for sequencing. I have a slide on that coming up here in a second, just to give you a broad overview. I've had a number of different roles as a part of this job. Done some technical project leading, focusing very much on the science of things, operation, transfer lead, really understanding kind of how that all interconnects with the actual business and being able to make stuff and send it out to customers so that it works for them. And finally, an actual kind of core team lead type position where you're actually managing other people. So, kind of, you have to understand a lot of the science and kind of what's going on, but really you're managing these other people who are taking on kind of those roles in terms of project development. Part of this development, it's really all encompassing. We go from product concept to launch. So pretty much everything that goes from optimizing all the way to producing and marketing material that we send out to customers. And it requires a lot of cross-functional work. So you get to see, in relation to this particular panel, we get to see a lot of the other career options that exist, working with quality assurance, quality control, operations, project management, just to name a few that I work with on a weekly basis. So I want to go to the next slide. I just wanted to give a quick overview of kind of just what we do at Illumina and what I work on specifically, kind of high level. For those of you who don't know, Illumina does NGS sequencing. And the basic workflow, how this works is shown on the top, where you take your patient sample in the form of saliva, blood, whatever it may be. You then prepare that DNA so that it can go onto the sequencer. This usually involves breaking it up into smaller manageable pieces and putting adapters on it. Once you've done that, you can put it onto the flow cell, put it onto the sequencer, read all those bases, and then the final aspect is analyzing, taking all those base calls that you have, putting them together, mapping them to the genome, and then ultimately, hopefully discovering important SNVs, etc., to help patients, determine therapeutics, determine what disease they may have, etc. So, and like I said, so the portion I'm working on mostly in terms of development was this library preparation. And this is just a quick overview of one of the projects that I worked on I thought it was really cool. And it involves this bead-linked transposome, or BLT, not just bacon, lettuce, tomato sandwich. And basically what this does is it uses transposomes, which you've probably heard of, and we utilize those to actually help us fragment up our DNA. And by putting it on a bead, it's a magnetic bead, so it's really nifty. So all the DNA gets attached to the bead. It cuts it up in nice, even lengths, because it's kind of spaced out on the bead. And because it's magnetic, we can easily watch the bead and get rid of all those reagents we use to ligate stuff onto the DNA, these adapters. And then we add indexes to this DNA that we've cut up so that you can tell the difference between your patient samples. You don't want to confuse the DNA. We do PCR to make sure we have enough to go onto the sequencer, and then we go ahead and sequence that and do our analysis. So that's just kind of a quick overview of what I work on in Illumina. So.
Okay, thanks. So now we next have Harvey Chen from Regeneron.
Hi, I hope you can hear me. I was having a few connection issues, so I've been dropping in and out. I'm signing in from my work computer now.
We can hear you.
Okay, great. So I'm dialed in twice now, so I just want to sign out on my iPad. All right, thank you for having me. It's a pleasure to speak at this panel. I'm going to describe my work currently in the Pharmacokinetics department at Regeneron. Just a little bit of background. I received my bachelor's in molecular biology at Princeton in 2004. I went on to receive my PhD in molecular biophysics and biochemistry at Yale in 2010 under Enrique de la Cruz, studying enzyme kinetics, primarily enzyme mechanisms and kinetics. And then I went on to do a couple of postdocs, moved to New York City, and did a bit of a meandering path, exploring different biological disciplines. So I started off at Weill Cornell in the biochemistry department. I did some cell biology, some microscopy, and quantitative kinetics, trying to understand the kinetics of cell biology a little bit, and then spent some time at doing a postdoctoral fellowship at Columbia in biological modeling and simulation, trying to tie together all those quantitative kinetic pieces into a coherent model that we could test in an experimental setting. And that was actually a pretty important transition in my career path to go towards a real, to really quantitative modeling type of career path. At that point, I sort of wanted to, I explored a few different career paths. It was the cusp of deciding between, can I actually build up a portfolio to go down the academic gauntlet for a faculty application path, or should I look at some career paths in industry or finance? Ultimately, I ended up in industry. I found a role at Novartis in New Jersey, and working particularly at the Novartis Institutes of Biomedical Research in the bioanalytical department, and got my first foray into pharmacokinetics and learning how to apply some of the things that I've learned during my my meandering academic path to apply towards a translational science type of role in a pharmaceutical setting. After a few years in that position and going through a few organizational changes, I was looking for the next sort of challenge, and found Regeneron was actually recruiting for positions within preclinical pharmacometrics. And when I interviewed there, I was really drawn to the sort of intersection between academic academic mindset and industry experience. So it's a real setting that you could actually apply a lot of the scientific mindset to a translational regulatory type of position. And I actually made this transition in 2020, right at the start of the pandemic. So I actually have not yet been in the office. I've been working fully remote since the start of my position there in April. And so I'm going on 1 year. Still haven't met my colleagues in person, although I've interacted with them numerous times in this Zoom setting. But it's definitely been a challenge to transition to a new role, but yeah, everybody has been so welcoming, and I've been able to apply a lot of the things I've learned from my previous experience to sort of catapult me forward in the current position. So I'll describe a little bit about my role in the next slide. But first, I wanted to just go over a quick 30-second overview of the drug development pipeline. The timing, I'm sure you guys are fairly familiar with, the whole process of how a drug gets made. It takes roughly 10 years and $2.6 billion to develop one drug. That's including all the failures, averaging in all the failures along the way. This information is a little bit outdated, so the numbers are slightly different now, but this sort of gives you the overview. Basic research on the far left side is sort of the pathway understanding what genes and what pathways are potentially important for particular diseases. And as you go forward, you get more understanding of developing a drug and how that drug is interacting with your pathway as well as the body. and less into the specific science, and so it becomes a little bit more of an applied science at that phase. So drug discovery is all about selecting that lead molecule, the lead antibody, and then weaning it down based on a lot of properties such as the efficacy in vitro and in vivo efficacy and toxicity in the preclinical phase. This is the phase that my group is actually primarily sitting in. So we interact with people on both sides of the light green, the yellowish side, and the darker green in the clinical trials, because we sit in that cusp of trying to translate the molecular understanding of the drug and its interaction with the body, and translating it from animal and in vitro data into what we anticipate in the clinic. So we have both a scientific and a strategic place here. And through interacting with the project teams, we speak a lot with the research, the discovery scientists, as well as with the clinicians as we're planning to hit that IND submission. The big white bar that is our major regulatory goal in our group. Then you go through phase 1, phase 2, phase 3. I won't delve too deeply into that, but this is, first in human trials, understanding the drug safety and properties in the body. Phase 2 is your proof of concept in the disease. And then phase 3 is your large scale clinical trials to really get those statistics to demonstrate efficacy. And that's the point at which we'll submit the NDA or the BLA. And my group, though we sit exclusively in the preclinical space, because there are animal studies that go on throughout the clinical phase that informs on the safety and efficacy as you go forward. We're involved in the regulatory filings, both at the IND step and the BLA step. And then for the last slide, just want to describe a little bit about pharmacokinetics and pharmacodynamics. These are phrases that I had not heard of until I actually joined industry. I knew what kinetics was. I didn't know anything about pharmacology. So when I first came across the job descriptions, I thought, okay, a lot of the applications seem to be things that I can handle, based on my background and my quantitative skills. So this is something that I'll go ahead and explore, but I didn't really understand what pharmacokinetics entailed until I actually joined industry, and I think that could apply to a lot of different roles. So if you go out and look at job descriptions, you'll find that they can be pretty difficult to translate into layman's terms, or even into scientific terms. And so a lot of the applications of what these roles are actually doing and how they interplay with the rest of the functions within pharma can be a little bit of a daunting process, but I'll try my best to describe what pharmacokinetics is here, and pharmacodynamics, which is the some of the more important aspect of the job. So pharmacokinetics is roughly what the body does to the drug, and pharmacodynamics is what the drug does to the body. The pharmacodynamics part is again looking at efficacy and toxicity, right? Effects on the body. Pharmacokinetics deals with how your body absorbs the drug, distributes it to the organs of interest, and gets to the adequate concentrations to interact with the pathway of interest. And that's sort of depicted on the bottom right side of the figure here. Pharmacokinetics shows the concentration of the drug in the body, and pharmacodynamics is sort of that curve, the EC50 curve that we're all familiar with from basic biochemistry. And we combine these two pieces of information to understand how we get the effect of the drug over time in the body. So this is the overly simplified way of describing it, but this is based in a nutshell what we do in the pharmacokinetics group. And we do all sorts of different experiments to tease apart this. And these are all things that we, or impact that we contribute to the drug development pipeline. So we help to develop the therapeutic index from the preclinical trials studies to what we anticipate would be efficacious doses in humans. And so again, we're speaking with the project teams to discuss what's the best strategy to get into the clinic in a safe way, and then escalate rapidly to an effective dose. And so obviously you need to understand what that dose response relationship is in the body. And we do that through various scaling mechanisms from animals to human. We measure the distribution to relevant tissues. For example, for the COVID antibody, one of the main calculations was what's the distribution of the antibody into the lung tissue, where it's interacting with the virus predominantly. We measure the fate of the drug, through its catabolism and excretion from the body, and then anticipate any potential drug-drug interactions. So all those things that your pharmacist will say, don't take the drug A with drug X because you'll get an overdose. So these are things that the pharmacokinetics group is involved with developing the strategy for in pharma. And the bottom left is sort of a depiction of the role of pharmacokinetics in drug development. So you can see this was back in 2012. Pharmacokinetics was actually the cause, or one of the primary causes of failure for drugs entering phase 2. And hopefully by the time we get to phase 2, we have a good understanding of the PK/PD relationship. And then by the time we hit phase 3, and PK no longer becomes an effect, because we've ideally weeded out any drug that would fail due to inadequate exposure in the target organs. Over time, the idea is to apply these quantitative methods to shrink that down to nothing as we get into the clinic. So hopefully we can get better understanding just through the animal and in vitro studies and quantitative measure modeling to predict the behavior of the drug, so that in the end only safety and efficacy will become the predominant causes of failure of drug development. So that's it from my side.
Okay, thank you. So next we have Jennifer Saucier. I apologize if that was not the right pronunciation.
You did great.
Okay.
Hi, everyone. I'm Jenny, and I'm a senior scientist at Sarepta Therapeutics. Harini, you can go to the next slide. So to give you a little bit about my background, one of the things that I show is this beautiful picture on the left of where I grew up in a small town called Millinocket, Maine. And those of you that aren't familiar with this mountain, this is Mount Katahdin. It's the topmost mountain on the Appalachian Trail. So I feel really privileged to grow up there. And if you haven't been up there, there's wonderful hiking, rafting. It's really a great place to visit, and I would encourage it. From there, I moved a little bit down the road to the University of Maine, where I actually majored in chemical engineering in the Honors College. But the one thing that I was hesitant about when I was in chemical engineering was that I still had a very big interest in medicine. And at that point, I wasn't sure if it was medical school or if it would be more the research side of things. So I really had to find a way to explore that. So as an undergrad, I made a point of taking some additional biology classes to be able to support that going forward. After I graduated, I still didn't know quite exactly what path I wanted to go. So I had an opportunity to become a lab manager at Yale for a few years, which was a wonderful stepping stone to really figure out what I wanted to do. And during that time, I had, was able to work with researchers as well as physicians, and really get a feel that, I felt like I fit well in the research side of things. This led me to actually continue to do a PhD at Yale in biomedical engineering under Mark Saltzman. Had a great experience working on the development of polymeric nanoparticles for various indications, but my primary research was on glioblastoma treatment. And during that time, I can appreciate Harini and Molly, all of your work. I was a McDougall Grad Life Fellow while at Yale, so had a fun time there. So from there, I actually decided not to do a postdoc, and that was a conscious choice at that point, and was offered a chance to join an early startup called X2 Pharmaceuticals. And at X2, I had a chance to work on polymeric nanoparticle development, so continuing some of the work I had done in grad school for new applications, primarily in oncology and fibrosis. From there, I worked there for a few years. Unfortunately, as with the majority of startups, we didn't get our later stage funding and had to close down. So X2 no longer exists, but it gave me an opportunity to re-examine what I wanted to do. And I had an opportunity presented to me to transition to gene therapy, which has many similarities in terms of the drug delivery field to the nanoparticle field. So I took on a job at Sarepta Therapeutics, where I'm a senior scientist in drug product development and manufacturing. So we can go to the next slide. So this slide here is actually, if you go back to the picture there, the slide here is a little silly, but the reason I wanted to put this in here, these were actually two of the people who supported me most during my graduate career. And this was taken in the Saltzman lab on one of those days where we were probably going crazy late at night trying to finish some experiments. But the reason I wanted to show this is just to tell everyone, you want to be able to find your support system. And I know this can be hard sometimes to find people who will support you either in industry or academia. Certain people have feelings in particular directions. So find somebody or some people that can support you and ultimately find something that you're excited about, because I know that I still have as much fun today as I did back then working and, progressing all of these therapeutics forward. All right, so I can't speak too closely on the particular work I am doing right now, but I wanted to give you a broader overview of my role as a formulation scientist. So you might hear the term pharmaceutical scientist, formulation scientist, drug product. They're all in a kind of a very similar realm. And throughout my career, I've had a chance to work at that early startup X2 and now work on both early and late stage development environments for programs. So Harvey, thank you for presenting that the nice overview of everything, because this leads in well. So in my role, I still work for, it's a growing company, but a very small company as overall. And I get a chance to get my hands in many of the aspects and many different programs in the pipeline. So really what we want to do is take an idea from the conceptualization and develop it to be safely used in humans. And obviously there's many, many steps along the way to be able to do that. And my role is to develop these novel drug delivery systems, such as the polymer nanoparticles that I mentioned, as well as my current therapeutics I'm working on, which are AAV gene therapies for numerous indications. And primarily I've worked on different oncology programs, fibrosis, and now numerous rare diseases such as muscular dystrophy. One of the basic things you need to understand to be able to do this on the formulation side is understanding the properties and chemistry of the therapeutics and how they interact with the body. We need to be able to understand the knowledge and have the knowledge to increase product stability and potency, which really optimizes the distribution of these therapeutics to the organs that you're actually trying to target. And what I mean by stability is, you want to have your drug not aggregating. You don't want to lose product as you're processing. You want to be able to provide the optimal dose and can keep your drug activity when it reaches your patients for administration. So one of my big roles in then what I'm currently working on is managing process development and formulation for these drug candidates. A lot of this includes characterization of the drug, so the actual molecule, so that we understand it a little bit better. We also have to understand the process to make the therapeutic, as well as the components used to make that drug. Lastly, one of the big parts of drug product side of things is understanding the final container closure. So this would be the vial that the product's in, and also the process to get it into the vial, which in itself was something that I in no way appreciated prior to coming to my current role. So we can jump to the next slide, Harini. So I talked a little bit about formulation, but really formulation is optimizing the matrix that your drug is in. This is by performing excipient screens and analyzing product stability. So excipient screens would be looking at different chemicals and ingredients that can be included with your drug, your active drug, to keep it stable over time. When you have a shot, for instance, if you're getting a vaccine, you notice that it looks like it's in a little jar of water, but it is, has many, many components in there that keep it stable over time so that it can make it from when it's manufactured all the way to when it gets to your arm, when it's actually administered as a vaccine. And there's a lot of work that goes into understanding what needs to be included to be able to have it in there. In another sense, formulation could be making nanoparticles and formulating your drug inside a nanoparticle so that it's stable over time. So there's many different kind of aspects that can be lumped in there, as well as just the general analyzing and characterizing these formulations so that you can understand how they'll perform over time. And one of the things that we work so closely with is many, many other teams to be able to do this. Some of my primary work was working with our clinical and commercial teams to design how the product will be administered to patients and ensure that our product is compatible with the administration components. This could be as easy as a syringe, it could be tubing, and you need to understand what happens between when you take the product out of the vial and it's administered to the patient. One of my other roles, along with the PD side and formulation and working at the bench side and driving those efforts, I also support external manufacturing for our drug product formulation, aseptic production, and characterization, which all falls under good manufacturing practices and GMP. So there's specific practices we have to follow to make sure that we are providing a drug that can be administered to patients. So I frequently travel to manufacturing sites. I work closely with our manufacturing sites to ensure that they are adequately making the drug in the way that we would intend it to be made. Lastly, one of the things that we do very closely is working on these cross-functional teams, and you'll hear the term CMC, or chemistry, manufacturing, and controls. And this is really a cross-functional team, including everybody from drug substance, drug product, regulatory, analytical, quality assurance, project management, to ensure that these therapeutics are made in the right way, that we're following all of the guidance to be able to submit adequate regulatory submissions and hopefully push these programs forward to our patients. And lastly, it's really just about applying creative and innovative approaches. You frequently have situations where you don't have a lot of time and you need to figure out creative solutions to be able to push things forward. So a lot of it's just thinking on your feet, using those skills that you're acquiring while you're in school, you're learning how to learn. You have situations where you need to make quick decisions. And we want to be able to make these decisions in a logical way, but also a very fast way to be able to push these therapeutics forward to patients. So that's pretty much what my role is as a formulation scientist, but I'm happy to entertain any questions once we get to the question and answer session.
Okay, thank you. And then last but not least, we have Tracy Mansfield, who is a senior director at Janssen.
Thanks, Rainey. Oh, am I getting an echo? I'm happy to be here today and talk to you a little bit about my career progression and what it's like working in industry. If you go to the next slide, I did mine a little bit differently so that you can see I've probably been in industry a little bit longer, a little over 20 years now since I left Yale. Thanks to all the panelists for really setting up the drug development process. I think that'll give some good insight about where I'm coming from. So I got my undergraduate degree. I have a BS in biology from Northwestern University, and I actually took a couple of years off before I came back to grad school, and I worked for a contract research organization. They're also called CROs. And these are companies that primarily support drug development by working on contract for different companies, doing work that can be outsourced. So, they'll develop, there are companies that are specific to developing assays. The one that I worked for actually ran clinical trials, found sites, and ran studies for different companies. And this was a company that evaluated contact lenses and contact solutions. And so they were in the device space. So I had some industry experience before I actually came to Yale to get my PhD. And so when I started, I actually thought that I would go back to industry after I got my PhD. And especially back in the 90s, industry was still not really accepted by a lot of the professors. They wanted you to stay in academia. And they kind of wore me down a little bit. And you can see there, I initially worked in a lab that was focused on oncology. We were looking for new tumor suppressor genes. And I was there, I had chosen my lab, and I was there for about 2 years when my professor decided to leave Yale. And I had to make the choice of whether I was going to go with him to his new university or stay. And I ended up staying at Yale, but that meant that I had to start my project over. So I was about 3 years in and had to move to a new lab. And I was lucky, I found a professor that I had read with during my qualifying exams. So it was a fairly smooth transition, but basically I was starting over. And at that point, it was a conscious decision where I said, okay, if this is what happens in academia, I think I might go back to industry and see how that goes for me. So I did my research in Rick Lifton's lab. We studied the genetics of hypertension and end-stage renal disease, primarily channels in the kidney, and that was a lot of the work that I did. And when I left, I also made a conscious decision. I felt like I'd almost done 2 PhD programs with the 2 different labs, and I went straight to industry, to a small biotech company called Keragen, which was located in Brantford, and it had actually been started by a postdoc at Yale. And at the time, they were primarily focused on expression technologies. So this was pre-RNA-seq technologies and chips where you could look at different gene changes in different experimental conditions. They had a PCR-based approach, and they partnered with a lot of large companies like Biogen and Glaxo, SmithKline at the time, and a number of different companies. And they were looking for alliance managers that come in that could understand the experiments that these big pharma companies were doing and kind of translate them to the people that were working in the labs and running the experiments, and then take the data and give it back to the pharma company and help them interpret that data. So I worked in that alliance manager role for several years. And during this time, around 2000 was when really the human genome sequencing heated up, and the company itself decided that they wanted to get involved in the really more drug development. And so they did a lot of sequencing and evaluating new sequences from the genome, and identified drug targets that were cell surface proteins that you could use to develop antibody drugs, and also antibody drug conjugates that were conjugated to toxins and could be delivered to different cells. And I was actually fortunate to move into what they considered at the time a project leader role, and really putting together these drug development teams and take some of these programs from the discovery phase, where we had just developed these antibodies, trying to figure out what they did, if they could have a role in disease or modulating disease, and take them into the clinic and really evaluate them. So I was at Keragen for quite a few years, almost 10 years, and we lived through what was called the biotech bubble. I don't know if you guys are familiar with that term, but it was a time around the sequencing of the human genome when the value of these companies that had absolutely no marketed products, were really not making any money. Their stocks just went through the roof. And so these companies were really flush with money and made a lot of investments. And then over the period of a couple of years, all of that value left. So companies that were small biotechs at the time ended up having to really refocus, lay off a lot of their staff. And we went through several rounds of layoffs and trying to figure out how to have a business model. And at that point, someone said to me, you might want to give Big Pharma a try. You're not so dependent on your compound working. There's a little bit more flexibility in moving between different types of roles. And so at that point, I actually moved to Bristol Myers Squibb, and I took a project manager role. So really, instead of being a project leader, which I could do at a smaller biotech company, I was brought into more of a support role for those same types of drug development teams. And I went back to some of the disease areas that I had worked on in my PhD, looking primarily at cardiovascular, and then focusing on metabolism and specifically diabetes. And after a couple of years in that project manager role, I actually moved into a different function, which was more of a clinical study director. So actually running some of those clinical trials. And I focused primarily on a drug that ended up being Farxiga, which is an SGLT2 inhibitor in the kidney that's used to treat type 2 diabetes. So really kind of fell back on some of that channel work that I had done in my PhD to help get that position, and actually used it to understand some of the patient data that we were generating. And I had the opportunity in that role to really be working on a late stage asset, so a phase 3 asset, which was filed with the FDA and other health authorities around the world, and went through what you may have seen recently with some of the vaccines, where you actually had an FDA advisory committee, which is a lot of work for just a one-day event, where the FDA really reviews and can ask any question of your drug development team before they decide whether or not they're going to approve the product. So I was involved in that, and then worked on an approved product, continuing to study the additional benefits of it. And during that time, Bristol Myers Squibb decided to actually get out of the diabetes development field and sold all of their diabetes assets to AstraZeneca. And so I had the opportunity to move to AstraZeneca in a new role, which was called Medical Affairs. And Medical Affairs is actually the group of people that support launched products and work directly with doctors to try and provide education, medical information, publications that really explain how to use the molecules, and then work closely with commercial to try and figure out how you can present scientific data fairly. We're the ones that are putting a lot of the labels on the commercials that you see about what you have to be aware of, and really putting some information in there. So I worked in that role. I had some deep clinical knowledge of the compound and learned a lot about medical affairs. And unfortunately, the position that I was in, the facility that was near my house closed, and they wanted us all to move to a different state. And at that point, I took the opportunity to move to Janssen, which is a part of Johnson & Johnson, and moved into a development team leader role. So it took me about 10 years to move from that project leader role that I had at a biotech into the same role at a bigger company. But I think there's a difference between the opportunities that you can have early in your career at a small company versus a big company, and what they're looking for. So if you can just move to the next slide, I'll tell you a little bit about what I'm doing now as a development team leader. So I think this was set up in terms of what a matrix team is and what cross-functional are. So I lead primarily early development teams. So that's the space that's coming out of discovery, where you have a molecule that you want to start testing in people, and then moving into those first in human studies, where we're really assessing safety and tolerability of the molecule and trying to pick the dose that you want to take forward. So cross-functional, I think if you look at some of the teams that I work with, I would have a pharmacokinetic scientist sitting on it, similar to what Harvey was talking about. I would have a manufacturing or a CMC representative, as well as a clinician a physician that would know about how to study this in people, someone from with regulatory background. A number of different functions would be represented on my team. And these are all what we call matrix teams, which means that none of these people report to me. So I have to use my influence in helping to lead them and bring them together as a team. So my role is really to create a strategic vision for the program, lead discussions, team meetings, help the team come to decisions. You can imagine that in an early stage, we're spending a lot of time trying to make decisions without having a lot of data. Is this tox finding or something that we've seen in our toxicology testing, is that going to be something that's going to stop the program? Would it mean that it would be harmful in people, or is it something that we can change the molecule and maybe make that go away? So we're making a lot of these types of judgment calls on whether or not this molecule has what it takes to move forward in development. So you're managing a fair amount of complexity, and also trying to align our stakeholders, which is just a nice word for people that in management that are making a lot of the decisions on whether or not these compounds are going to move forward. So like I said, we're aligning with leadership. We're trying to make go, no go decisions and also managing timelines, budget. There's always constraints on what we can do and how quickly we can do it, and trying to get all of that together. So this type of job, I've just listed some of the job qualifications here, but what I really like about it is that you're sitting in the middle of a lot of data that's coming in and trying to make these trade-offs. We call it, benefit risk assessments to make the best decisions for the program. But it does really, you do come out of having some specific area of expertise, and then people that have strong communication skills, some leadership experience, and some disease area expertise are going to really do that. And then people do really specialize as to whether you're leading teams that are very early in development, discovery, early development, kind of looking at that phase 1, phase 2 space, or later in development where you're trying to do your really large clinical trials and working with regulatory and commercial to try and get a managed product. So focusing on those areas of development too.
Okay, thank you so much, Tracy. And thank you to all the panelists. Those were really interesting slides, and it was really interesting to hear about all of your different paths from your PhDs at Yale to what you're doing now. So we've had a lot of questions in the meantime, and unfortunately we don't have a ton of time left. So I've tried to kind of take some of the questions and combine them together, but I think unfortunately we still won't get through all the questions. But I'll start out with one question, and after I pose it, I think let's try having each of the panelists just speak up, not necessarily in any particular order. We'll see how that works, and if that proves to be too awkward via Zoom, we can also next time try sort of asking people turn by turn to answer. So the first question that I have is, what does career progression look like in your career and in your industry?
So I can start. Oh, I think Tracy's talking, but it's on mute.
Oh, sorry about that. Yeah, I'm happy to start. You saw my career progression. I think that I have taken the pathway where I've really tried to focus on different disease areas, being a bit of a generalist, moving between clinical leadership, some alliance management. I think that's one way that you can go is trying to get different types of experience following different aspects of drug development or research that interests you. There are other people that stay specific to one area of development. So if you're a pharmacokinetic researcher, that you work your way up, staying in that specific function, but move up from a scientist to a director to a vice president and really move that way.
I can add on to that a little bit. Kind of high level, how I have kind of like to think about the options, because there really are a lot of options at these companies. Is you can go, a very technical route. You can go kind of a management route, and you can go kind of a leadership route, and they're all kind of intermingled to some extent. But you can imagine that there's some scientists, you go in there and they like being in the lab, they like crunching the numbers, they're looking at the data and analyzing, and you can progress, all the way up through VP level, going that route. You don't have to, necessarily manage huge teams necessarily, right? And then there's management where it's like your goal is you're running people. And some people really love running people, and then that's one. And then there's kind of the leadership, and there's a lot of management in terms of leadership, but it's less about the people and maybe more about the projects and the products in the future of the company and strategic thinking and those kinds of lines. And so, and that's, I think, true in a lot of different aspects. I'm in R&D, but I see similar trajectories, high level at least, if you go into operations, if you go into quality and stuff like that.
I think the only thing I can add to that is considering the large company versus small company. I think probably Tracy touched on this a bit, right? You do have a lot of opportunities to see what else is out there, right? Drug development is a very complicated, team sport, and there are tons of different functions involved, right? So in a large company, you do get the opportunity to see all of those, explore all of those, and interact with scientists in different functions. In a smaller company, you're kind of doing everything on your own and learning as you go. And each one has its pluses and minuses, right? Sometimes a large company has a bit of bureaucracy, and so that vertical path of scientist to lab head to director is the default, right? Maybe similar to an academic path, but there are plenty of people who make these lateral moves, and you explore the different functions and you find a good fit, right? So you find that good skill set, interest, and company need fit. And that's something that you get an opportunity in a big firm. I've seen a middle-sized company like Regeneron actually has, it sort of has a little bit of the best of both worlds, where you can see the verticality, but you can also interact very seamlessly with the different functions. So that's something that you might want to consider as you're selecting between small and large companies.
Yeah, and just to add to what Harvey said, I mean, I've seen both at this point. I was number 6 person hired at the startup I worked for, and many of the people who were there were at the executive level. But there were many times where when you have so few people, everybody has to do everything. So you get to have a chance to do the really menial tasks that nobody wants to do. I remember my vice president and I fixing an ice machine during one of our first few weeks on site because it broke in our laboratory. However, at that same time, I had a chance, I was managing a full team in the lab. So it depends on where you are and what you're really looking for. And I think part of it is asking for opportunities. If there's things that you're looking for, even at any role, make it clear kind of what you're looking for and seek out those opportunities and just take advantage of them when they come along.
I just want to add one last thing, and it might be part of other questions as well, but like one thing I wish I knew before leaving with my PhD was how many different opportunities there are, right? You're in academia and you're like, I do science, I do lab work, and that's kind of all you think about. That's all I thought about. But there's so many options out there, and there's so many opportunities. So no matter where you go, make sure you're open to working in these different opportunities that people are talking about.
Yeah, I would add on to that I think things like this are so important. I heard all of the panelists today use, there's a different jargon that we tend to use in industry than what you hear in academia. So you'll see these job postings and you'll think, project manager, what is that? That's a very defined job in industry. So, reach out to people that in industry. They can help interpret some of this for you and really give you a better idea. Because it does, I think, I remember looking at job listings and trying to figure out what these things meant. It's not intuitive for some of these jobs, what they are.
That was a great plug for informational interviews. We have workshops at the Office of Career Strategy on informational interviews. I think Karini runs the informational interview workshop, and that's a really great chance to ask questions like that. And in fact, I had one with Jenny in the fall, which is how we got to know each other. So it can be incredibly helpful to parse through some of that jargon. And actually, Tracy, someone had a follow-up question to your answer on this question. You talked about having strong communication skills, and they were wondering, what do you mean by that exactly? What does that look like? Because it's something that comes up on job descriptions a lot.
So, you can approach that a couple of different ways. I think when you're in the lab, you're working in a lab, but you have your own project and you're really kind of doing your own thing. You're not immersed in a part of a team the way that you are in industry, where you're working closely with a lot of people. And so it does become very important that you can communicate with the other people that are on your team. I always tell people, people don't go to graduate school because they want to communicate. They don't want to they want to do science, right? That's why you're there. And so having those skills where you can reach out to someone that you don't know and have a conversation and get your point across. You have those presentation skills where you can get up in front of the CEO of the company and give a pitch and have them understand what you're asking for and why your program maybe is spending more money than it's supposed to, and get them to support it. Those are the type of things I don't, at least when I was back in graduate school, they weren't things that were taught. I have heard that there are opportunities to learn those more now, but being able to have those conversations are very important in industry. I think my advisor used to travel a lot. And so he would walk into the lab and really you had to be able to express to him in about 2 minutes where you were with your project, if there was anything that you needed from him, what we call an elevator speech. And I think that having started in the lab, being able to explain what I was doing in a succinct way and get his support kind of sets you up in building those types of communication skills that you need. So start there, right? Practice your project, be able to explain it to your mom or your family member, what you're doing and being able to communicate. That's a good place to start. And then you would just build on that.
Thank you. We also have a workshop on telling your research story. So just thought I would plug all of our OCS workshops during this panel session. So I think we probably only have time for one final question. And oh dear, sorry. In a lab room where there's an automatic light. So I apologize, this question's a little bit of a behemoth, but it we're gonna take you back to your transition from your PhD or your postdoc into industry. And a lot of people had questions about that transition. So I'll list off a bunch of questions. Feel free to answer the ones that are interesting to you or you feel like you have something to say about. So some of the things people were wondering about that transition were what factors did you consider when choosing between academia and industry? How did you prepare for the applications? And do you have any recommendations to students that are looking for a position similar to your first position in industry? And especially any tips regarding skills or knowledge that might not be naturally developed or incentivized during academic training? Similar to kind of what Tracy was talking about with communication skills. So if there's anything along those lines as well.
So there's a lot there, but I can I can speak to a few of those points. So one of the reasons, I was in academia working there for a period of time, even prior to starting grad school. And so I was obviously had a chance to be in the environment a lot, see how things were. I also was familiar with people in industry. So I did know people that were in industry at the time, and also had a chance to evaluate kind of what they were doing. But for me, it was kind of a gut instinct having had a chance, while I was there, I had a chance to work on grants. I had a chance to develop therapeutics at the bench side in the early stages. And it was something that I wanted to be more on the translational side of things, further along in the pipeline. So I consciously made a choice that I thought I'd be better off in industry, and really tried to cultivate that by talking to people in industry, finding out what they did. Ultimately, my first job, I have to admit, kind of came to me. I didn't have to, somebody had reached out. This is my plug for my grandfather. Always used to tell me it's you have to be good, but you also have to know people. So in terms of what Molly was saying, know people, reach out to people, because your networks will ultimately serve you a wonderful purpose. I wanted to think that it was just my qualifications that would get me to my jobs, but I've ultimately learned that part of it is just that networking, keeping in touch with people reach out and just ask questions, and don't be afraid to do that. I think most people are very receptive to helping people out, especially early in their career. So my first job, I did kind of fall into that, and it was a perfect position. Luckily it worked out. But I did with my second job, as I said, when the company went under and it didn't any longer exist, and we only had about 3 weeks of runway. to find something new, I really had a chance to reevaluate and did go through the full application process. And at that point, again, it was reaching out to my network, using LinkedIn. Even though if you're in grad school, undergrads, get on LinkedIn, set yourself up a profile that can be updated regularly, and reach out to people and make those connections so that when the time comes that you do need them and you need their help, they'll be there for you.
Definitely speaking to people is crucial. And I think one thing I've learned both during the interview process as well as sitting on several panels, and which reminds me, check out Regeneron. We've been hiring quite a bit recently, and I was just told my group is hiring 3 more people this year. So if you remember anything about PK and looks interesting, please check it out. But what we typically look for are, these tend to be more behavioral interviews, right? Once we see what your skill set is on your resume, we do drill into to make sure that you do know those things, but we look more into how you communicate and how open you are to communicating with people of different disciplines, because again, this is a team sport. So we need you to be that face of our group as well. So you're going to be out there and interacting. So when we interview, we're looking for people who show that genuine interest and humility to learn new things, and not go in with the assumption that I know everything in my field, and that's why you should be hiring me. We're looking for the communicators, the people who can tell their story in a succinct way, or tell it to somebody who's not an expert in your field, and have a little bit of influence skills, right? When you're dealing with uncertainty, as drug development tends to be very early on, especially, you have a ton of uncertainty, and to be influential, you need to be able to speak your mind and find ways to persuade people who you don't have, who don't report directly to you, who have other motivations and whatnot. So those are some skills that we're typically looking for and tend to rise to the top when during the interview process.
Yeah, I second that, right? I think the networking is so important. I tell my own kids who are in college that every job I've had since graduate school was helped because someone I knew recommended me. It's much more unusual to get a job that you're just cold called for, and their companies are always looking for someone who can vouch for you. So even if it's someone that you don't know very well, that you can reach out to and have them put in a good word for you, it's very important. Someone on the chat asked about industry internships. I think they are helpful. We definitely hire people that have come in through the internship route. It's a newer role, especially for people that are in school. But if you have the opportunity to do it, I think it's not just good for the company, but it's good for you to really get an inside view of what it looks like to work for a company, right? Do you like being in a function and doing the kind of work that you're being asked to do? I think it is very insightful to help you decide between career paths and which way you want to go.
And I don't have too much to add to everyone else. I just wanted to kind of reiterate, just one, the networking, but then on the other side, like once you have your PhD, people basically assume that you're capable of thinking and you're defending your thesis. So it's really a question, like Harvey was mentioning, it's about teamwork. Are you going to fit in at the company? Are you going to be able to do the non-science things and work as a part of that team? And that's a big part of what we look for when we're hiring people.
All right. Thank you so much to our panelists. I think we're going to wrap up now since we're at the top of the hour. But we really, really appreciate that you joined us tonight. You all had amazing advice, and it was really interesting listening to all of your stories. And just to make another plug, we have another workshop in 2 weeks on the job application process, which we kind of touched on a little bit here, so you can think of that as a teaser. So good night, everybody, and thanks again to our panelists.
Thanks. Good luck, everybody.
Thanks for having us.
Bye, everyone.
Bye.